Hi, everyone. So thank you all for joining. We're going to give everyone a couple more minutes to join the Zoom session before we get started. So we'll start in a couple minutes. All right. Thank you all for joining today for the April edition of Life RCN Seminar Series. Our invited speaker is Dr. Picard from the University of Nevada, Las Vegas. Her research areas of interest involve geomicrobiology and biogeochemistry, and she has a heavy focus on the study of sulfate-reducing microorganisms and their interaction with anoxic environments and at the oxic-anoxic interface. So we'll let Dr. Picard present, and then maybe with the last 15 minutes today, we'll open the floor to questions. And with that, I'll let Dr. Picard take over.
Good morning and good afternoon, everyone, wherever you are. Thank you for the introduction. All right, so let me share my slides. All right, can you tell me if the slides are okay?
I can see them just fine.
Okay, perfect. Thank you very much. Okay, so let me just remove those panels. Okay, we're good. All right, so I will, so first of all, I would like to thank the organizers and Katie Rico for the invitation to this meeting. It is a pleasure for me to try to present my work in a different perspective. So I'm mostly interested in, I want to say, in the details of those interactions. Just understanding the interactions between organic and inorganic material. The organic material being the microbes, so which are alive, and the minerals. In this case, we're going to talk about iron sulfide minerals. So I will first present my system of interest, and after that I will go back into more the astrobiology context for this. So we do have, I'm interested in sulfur-reducing bacteria, and this is a, so here we have an illustration from a review on sulfur-reducing microorganism by Robert et al. So what they call the metabolic principle of sulfur-reducing bacteria. So these organisms are tremendously important on modern Earth, even if they live in anoxic environment, which are now limited in volume. However, so the principle is that they take up organic compound that they oxidize, and they couple this oxidation to the reduction of sulfate, or sometimes elemental sulfur, sorry, and then they produce hydrogen sulfide. And they also take up some of this organic compound, nitrogen compounds, to produce more biomass. So these organisms currently drive the modern cycles of sulfur and carbon based on that metabolism, but also of iron and other metals. And currently at the surface of the Earth, they produce almost all the sulfide. I think it's been estimated to 97% of sulfide produced at the surface of the Earth is actually from sulfur-reducing microorganisms. So I'm only talking about bacteria today, but there's other archaea that also produce sulfide. So yes, the rest, the 3 remaining percent of sulfide is produced at hydrothermal vent. We have a significant amount of sulfide produced. And then here's where the iron comes into play, is that whenever there is iron in the system, iron 2 will interact very quickly with sulfide and then produce iron sulfide minerals. So I will start with the most famous one, which is the pyrite. Here's a nice example from a mineral collection. And it is, this is actually an example from hydrothermal environment, but this is the most stable, also the most interesting for geoscientists to reconstruct the past environment, also modern environment. What I'm interested in here mostly are what we call the metastable minerals, iron sulfide minerals, so mechanoite, FeS, and greigite, Fe3S4. So mechanoite doesn't have this nice, those nice crystal forming and growing. So what I'm going to show you here as an example of the mechanoite is basically what floats in my cultures of sulfur reducers grown with iron. For greigite, there are a few interesting examples. In the living organism, we have this deep sea gastropod, basically forming a scaly foot covered with greigite and sometimes also pyrite. You might have heard of magnetotactic bacteria, which produce magnetic minerals. So the most known is magnetite, but they've been shown also to produce greigite. So you can see on those images that you have the minerals that are aligned in the bacteria, and then this is a close-up look. So this is a control formation of mineral, and they are supposed to use those minerals to align themselves or navigate using the magnetic field of the Earth. And so the final example that I have for you is just taking that same culture and placing a magnet next to it. So whatever is floating here or stuck to the glass vial is actually the greigite. So now I'm going to try to put this back into the context of this seminar series. So I was looking at the details and the previous speakers. So I try to think about those interactions in terms of biosignature, but as I said, I'm very interested into the basic details of the interactions. But if we look back at the timeline of Earth formation, potential start of life, we had on Earth at least. So if we think that life started around 4.4 billion years ago, or even before, we basically had Earth or Earth surface or subsurface that was anoxic for at least 1 billion years. And the sulfur-based metabolisms are thought to be very ancient. So we have this time where potentially sulfur reducers evolved and thrived all over the place. And if we look at after the time oxygen starting to be produced biologically and took over in the atmosphere, we still have a significant amount of environment that are anoxic, that are important for the global biogeochemical cycles on Earth. They're limited in volume now, but however, they still remain significant in terms of numbers of the mass of sulfate, of sulfur, iron, carbon that can pass through the cycles. Also, something that is very interesting for the start of lifetime in relation to iron sulfide minerals. So there's this iron sulfur world hypothesis that shows that potentially iron sulfide mineral played a catalytic role in the origin of life by providing catalysts to make complex molecules from some simple molecule. But there's also this proposed structural role where the first membrane would have been iron sulfide minerals bubbles instead of starting with lipids. So in this sense, my kind of claim for this overall talk is that interactions between iron sulfide minerals and microbialism, more specifically sulfur-reducing, is quite ancient. And as I will tell you in the remaining of the talk, is that I think that those interactions are very unique, and they're different from interactions between microbes and other types of minerals. So this is another illustration from that same review by Aude and Lisbeth. So this is from 2012, so it's a little old now, but still, this is a representation of the early Earth, and potentially because we had this global distribution of anoxic environment, there was no oxygen, we had the potential to precipitate iron sulfide minerals in a variety of environments. So we have the active hydrothermal systems on land or in the deep sea that were producing, just releasing a lot of metals that could precipitate with sulfide. And there was, although the concentration of sulfate were much lower than in modern time, we still had sulfate, potentially elemental sulfur, that could be biologically reduced to produce sulfide. So a number, if we look at modern environment, we would have iron sulfide mineral precipitation, mostly in the subsurface and sediments, but in early environment on Earth, we would actually have precipitation all over the place, including oceans. So when I started this work, I basically came across a study that was kind of going into the sense and into the direction of the idea that iron sulfide minerals might actually be beneficial for sulfur reducers and maybe microbes in general. So I had basically worked on the effect of sulfur reducers on iron sulfide minerals, and then started thinking about the question the other way around. So what is the role of minerals in the life of microbes? So while writing around, I came across this paper that looked at the potential for iron sulfide minerals to help electron transport to the cells to produce energy. So this is an example of a sulfur-reducing bacterium called the sulfur-vibrio vulgaris, which has been grown in a medium. So with sulfate, the electron acceptor, lactate is the organic compound that they oxidize, so producing energy in the presence of some iron too. So what they show is some precipitation. So it's not really really massive, but you have minerals on the outside. It seems that they are like particles in the periplasm, and so their model, the idea is that if they measure, there's a potential for increasing electron transport to the cell to produce energy when they have those nanoparticles precipitated. So basically, there's more, because iron sulfide minerals are semiconductors, there's a potential for an enhanced transport of electrons to the cells. So it seems that that is what they were seeing. So they used the system of electrodes, and so this is cells. So no current density. The current density doesn't change over time in a sterile environment, in an environment without the minerals. But when they are minerals, then current is basically enhanced. The current transport, electron transport is enhanced from the electrode to the cells. What they've seen is because this getting more electrons allow the bacteria to produce more energy. There was also in the culture within the minerals, they were seeing more assimilation of nitrogen compounds, which goes hand in hand with the idea that if you have more electrons, then the bacteria can produce more energy and then can produce more biomass, or at least assimilate more organics and nitrogen to make new biomass. So starting from here, I have kind of the work organized in 3 sections with 3 questions. Basically, you just like have this arrow going in one direction, and then the arrow will go in the other direction. Or so the first question is to understand if the biogenic iron sulfide minerals are unique, in a sense that do we have an influence of sulfur reducers on the physical property or composition of the iron sulfide minerals, and this is relevant when we look into biosignature in iron sulfide minerals. And after that, so you might guess that there are some unique properties, and so to try to understand this unique, how the microbes can produce iron sulfide minerals with unique properties. And I have to point here that it is not a controlled mechanism. It is just the, as I showed in the beginning, those bacteria produce sulfide. If iron happens to be around, then the minerals will be formed. But then we have to understand the interaction, as I said, the interface between organic and inorganic materials. So is there affinity, is there repulsion, is there anything in the properties of minerals that allow them to maybe be close to the cells or away from the cells? And finally, looking at this interaction, as I said, I look at the problems from the other side and looking at the potential beneficial aspect of those iron sulfide minerals for the life of sulfur reducers. So I'm going to now describe the experimental setup to try to answer all of these questions. So I have those different conditions, and these basically allow me to answer all those questions together. But after that, I organize. I will show you results based on those 3 questions. So we have I want to say the most common conditions that I'm looking at, which I call biotic, but there will basically be other biotic conditions. So in this case, we have a medium, which is very common medium to grow sulfur reducers. We have sulfate reduction coupled to lactate oxidation that allow the microbes to produce energy and then produce biomass. And then we grow them with iron 2. And I've used different sulfur reducers, but most of the result I'm going to show you is for this strain called the sulfur-vibrio hydrothermalase that has been, that I picked specifically because it was isolated from a deep sea hydrothermal vent. So it was basically used to being in contact with sulfide. And then as soon as the bacteria produce sulfide, you can see the medium darkening. So it's a very easy indicator of growth in sulfide production because it turns black and becomes more and more opaque, as I showed you in that first slide. Then we have the kind of control condition where we do have exact same conditions, but instead of using live microbe, we use the cells that have been previously gamma irradiated with gamma radiation. So that basically kills the cells, but we still have this availability of the cell surface, and I will show you in the next slide why this is important. So the cells are not active anymore. And then we can check that first. We add the cells to the medium with iron, but they do not produce sulfide. So we have to add sulfide to the system eventually to have iron sulfide mineral precipitation. My abiotic control are numerous. We use the same medium, and then without anything, actually even without lactates, we add sulfide, but we have all the conditions possible. So the medium is a seawater, is a simulation of seawater, but we did with the lactate that is used as a source of carbon and energy for the microbes, but we also use without it. And then we tested a bunch of organic molecules to figure out the differences between the biogenic condition and the abiotic condition. So again, we have to add sulfide because there's nothing that will produce sulfide in the system. And we also tested a number of rates of addition. So obviously, it's very difficult to reproduce that diffusion happening from inside the cell to outside the cells. But here we tested different additions. So just adding all the sulfide at once or adding drop by drop, trying to reduce the rate. And so my second second kind of biogenic conditions are a little different from the first. So I grow the microbes first without the high concentrations of iron, and then I add iron. And I will show you quickly in just a second why we do that. These are microbes that do not like oxygen. The minerals are also very sensitive to oxygen. So we basically do everything into the anaerobic chamber, and we have we transport the sample to the instrument in anoxic environment. We just use mason jar, which are really good at keeping air. They're really airtight, at least for up to a few weeks. And then we use a number of methods to look at the properties of minerals. So just to go back to the explanation about why we have those two biogenic conditions or biotic conditions. In the first case, where we grow the bacteria with iron, we have the expectation that iron will bind to the cell surface. So most bacteria have a surface that is negatively charged because of the deprotonation of functional groups in organic compounds. So in those conditions, we are expecting the cells acting as templates, basically, where we have first iron binding and then the sulfide being produced inside the cells and then diffusing out. So I mentioned that we have also the dead cell control. So in this case, this is why we put them in the medium with iron, hoping that even when they're dead, they still have this charge. And and then in the case of the dead cell, we would add sulfide. And then this other biogenic condition that I mentioned, so we first have the cell produce, grow, so they produce sulfide, and then we add iron. Because the reaction, the kinetics of precipitation of iron sulfide, of reaction between iron 2 and sulfide is very quick, there would be no chance for iron to bind to the surface. So basically, in this case, we have what I call the templating effect. Here we don't have it. So we are expecting differences between these two cases. And basically, for the abiotic condition, we try to use anything that would be relevant for the system, for like a prebiotic system. So simple molecules, we also use, I didn't put it here, but we had mixture of amino acid, and then we have more complex mixture like the tryptophan, the yeast extract. Lactate is also what we use to feed the microbes. So we incubate all of that in the medium with iron, and then we add sulfide. And here we don't, I didn't really have expectation about what we could see, so I'll just mention that afterwards. In that first part, so now I'm going to present what do the sulfur reducers do to the iron sulfide minerals to try to not necessarily convince you, but show you that the biogenic sulfide minerals are unique, can be unique, and that they could potentially be used as biosignature for the environment on Earth, and maybe looking for life on other planets. So the most appropriate methods to look at the mineralogy of sulfides is actually X-ray diffraction. So if the minerals are crystalline, we do have those planes that are parallel to one another, and we can have diffraction of an X-ray beam onto those planes. So depending on the type of detector that we have, we can see that as a 2D pattern, or we can have a typical diffractogram. Having this showing back up. Okay, so this is an example of what we see when we look at lots of different conditions and time point of iron sulfide minerals produced by in cultures of this sulfur vibrator hydrothermal is grown with iron. So we have here our McKenna-White and Greigite standard. So it's showing us the expected peak and relative intensity. In none of this culture we saw pyrite, and I think that the reason is that pyrite needs often is produced when we start with a source of iron that occurs as iron 3. So in this case, I did all the experiments with iron 2, so we see mostly these two minerals. So you can see first, so I've identified the peaks in those spectra. But what you can see is that in some cases we have this fine peaks with high intensity. Actually is a way of showing that they are more crystalline, and then they're forming larger crystalline domain. And then we have these cases where we have broad peaks, very small intensity, and this is an indication of smaller crystalline domain. So what we have here is that in the biotic condition, so with live cells that grow with iron, those dead cells that are incubated with iron, and then with sulfide added afterwards, we see McKenna-White that is very crystalline. And then after 5 months, we start seeing Greigite. So 5 to 6 months, it's been reproduced by others. And it only happens when we start with those live cells grown with iron 2. And all of the other conditions that I've shown you is that the abiotic condition with different rates of sulfide addition, the abiotic condition with organic molecules, and those biogenic conditions where we add iron later, we do not see, we see much smaller particle size. And so tell us here that the larger crystal is due to this templating effect where iron can bind to the cell surface before it interacts with sulfide. And then we have a role of microbes, of live microbes. We still don't know really how, but that drive the transformation. So there's no Greigite at all at any time. I looked up to several years. There's no Greigite in all of these conditions. And so, as I said, we do everything in anoxic condition. Everything is comparable. So in all conditions, I can say that the microbes, the live microbes, the originally live microbes are doing something to produce Greigite from McKenna-White. So this is a plot of, so we can extract quantitative data from the spectra. As I said, the crystalline domain can be estimated. And so this is McKenna-White in all those different conditions. And you can see that so this is the crystal structure of mechanoide, which is produced in all conditions. So I'm showing here the particle size along the AB axis. And so we can see that when we have this biotic mechanoide precipitated in cultures grown with iron, so whether or dead or alive, it's pretty large and about twice the size as the abiotic or this biotic where we add iron later. So we have this templating effect that produces a larger particle size. And so, as I said before, we tested a number of different rates of addition in the abiotic experiment to figure out if it was due to a diffusion effect. But again, it doesn't have to do anything with the rate of sulfide addition, because in the dead case, we add sulfide in the same way as we added in the abiotic condition. So we can also calculate the greigite size, and it occurs to be, it happens to be in the same range of size, which is consistent with the pathway of production of formation of greigite. Greigite is actually formed by the solid state transformation of mechanoide. So that makes sense that when we have this biogenic greigite, it is in the same size range as the mechanoide. And as I said, there is this potential role of metabolic or residual microbial activity to transform mechanoide to greigite. So I should say that in greigite, iron is in the occurs as iron 2 and iron 3, while in mechanoide is only iron 2. So there is an oxidation required for that transformation. And obviously, the sulfur reducers do not get electrons from iron 2. But yeah, I'm thinking about some kind of residual activity that would lead to the oxidation of some of the iron 2. So still in progress. I still don't have the answer to explain that mechanism, but it's been observed in other studies. So then I started trying to do some higher resolution work more recently. So we know they're more crystalline. So this is a biogenic mechanoide produced in a culture grown with iron after 5 days. They already reached this very large particle size. So we can see nice particles, and they're all really crystalline. So we can see those, you can probably see those lines, parallel lines. These are basically with a specific distance between one another. So if you look at that crystal structure, it's basically showing those lines here. So they're basically, the minerals mostly grow in the AB axis. And so what we see is the distance, the C distance between those parallel planes. So supercrystalline material, they diffract a lot in the under the electron beam, and this is older samples where there's already there is a lot of greigite, some mechanoide left over, so the transformation here is pretty slow, but very crystalline. Again, we see those very nice lattice fringe. So I didn't show the result here, but so we have the biogenic minerals that become crystalline very quickly. And so for the abiotic minerals to become crystalline, so they don't even reach that level of crystallinity after a year in abiotic conditions. So we have something in that the microbes promote, is just making them supercrystalline very quickly. I started using synchrotron spectroscopy. I'm still working on the data analysis, but you can see this is the abiotic mechanoide after 2 weeks. So the spectrum here, the black spectrum is the one after a year. They're pretty similar to one another. And here is the biogenic one after a month, where we're supposed to only have mechanoide, but you can see how different it is from the abiotic mechanoide. So if we looked, when we looked at the X-ray diffraction data, we could see that basically the diffraction only tells us about the particle size. So it is mechanoide in both cases. There's no indication of a change in the cell parameters in the crystal structure, and here we see differences. I cannot still really explain what the differences are, but obviously the early time where we're supposed to have only mechanoide, it is very different from the abiotic mechanoide. And late time, obviously, when we have gregaide, it's even obviously even more different. So conclusion on that side is that on that specific part is that the biogenic iron sulfide minerals are unique, and this is important if we want to use those biogenic sulfide as biosignature. There are obviously other things that we can measure in those minerals to make them even more robust biosignatures. But in this case, I'm only looking at this specific crystalline parameters. And now I'm going to look into the interactions where we look more into the composition of the minerals. So to understand micromineral interaction, we can use higher resolution microscopy. These are cells imaged by scanning electron microscopy. So if you look at the cells grown without iron, they look pretty smooth, but they also look very smooth when they are grown with iron. So I had worked with other systems before where cells, when they are encrusted, it's very obvious. You can see the mineral spikes at the cell surface. But in this case, I could not make any conclusion using SEM. So it could be that they have no minerals on them or very smooth minerals. So I decided to use actually transmission electron microscopy, which is a method that has some bias, but we did the control for the abiotic minerals just to make sure that the structure that we see in biogenic conditions are unique to biogenic conditions. But I will go quickly through the preparation. To preserve cells, you have this first step of strong chemical fixation, which is indeed using aldehydes to cross-link proteins. Proteins make up 50% of the dry mass of a cell. And then we use a secondary fixation with osmium tetroxide, which stabilizes lipid. So in this case, we kind of wash off all the material that is not associated with protein. So all the potential polysaccharides, things that would be around outside of cells, kind of washed away. But still, we preserve pretty well the cells. Then we need to do some heavy metal staining because the cells are mostly made of water or light elements. So if you look at cells under a microscope, they're almost transparent. So we increase the contrast by using uranium, for example, or non-radioactive alternative like lanthanides. Then we have to dehydrate the sample because electron microscope are under a very high vacuum. So we need to remove the water. So we exchange the water with serial dehydration for ethanol. Then we exchange ethanol for acetone because acetone can dilute resin. So then we slowly replace acetone for resin. Eventually, the cells, everything's completely filled with resin. We cure the resin, we make thin sections, and then we're finally able to image. So as I said before, we have this expectation that we have this templating effect that could also actually lead to incrustation of cells, which is what we see. So this is all the work where I've realized now that I was lucky to sample at the right time. So this is a 1-week sample that shows cells completely encrusted. On the left side, this is the comparison of cells grown without iron. So you can see this thick crust. So my assumption here is that cells were encrusted forever because I had to work with other systems where cells encrusted in oxides are actually encrusted forever. So that was my first assumption. So this was done before I was at UNLV. So I looked at this again. So this is reproducible. And we looked at the time at which it becomes encrusted. So it starts growing in a medium with iron, start producing sulfide, and then we have this templating effect that also leads to the formation of crust around the cells. But then eventually I looked at cultures of different time after 2 months, 7 months, 4 years, and many other in between. And then we just realized that the crust was gone. It was impossible. Maybe one occasional cell completely encrusted, but we couldn't see crust anymore. So looking more into all of the images collected, we could see that a lot of those structures, which look like basically peeled off crust. Everywhere we see that, and we also see this kind of gradient where when we have a mineral aggregate, it's not a good example here, but when we have a mineral aggregate, we have cells next to the aggregates that are encrusted, and then encrustation kind of goes away with the distance from the aggregate. Because basically, it looks like even the minerals are kind of attracted to that big aggregate, and then the cells just get rid of this crust somehow. And so something very interesting for the biosignature kind of ideas is that those so you can see those crusts like kind of like an orange peel that kind of end up stacked together. And so, as I said before, the preparation for electron microscopy is pretty harsh. So we tested the abiotic minerals with the exact same preparation, and then we see that they are very different. So this is a similar magnification. So we only see those super cool structures of stacked crust, what I call stacked crust. So we only see that in those biogenic conditions when the microbes are grown with iron. So trying to understand how the microbes shed minerals, it looks like the crusts are pushed away from the membranes by extracellular membrane vesicles. So this is something that we see in the cultures without ions. You can see those vesicles being formed, and they are just like popping out of the cell. So this is a very common process mechanism or process that happens in cells, whatever happens, whether iron is there or not. And then we can see in cultures with ions that we have lots of those vesicles being produced, and then close to minerals. We also see encrusted bubbles like this. So it's kind of maybe a way to explain how they get rid of it. So, but very interestingly, the crusts go away and form those very big structure, but you cannot see cells encrusted after a short period of time. So what I showed you is basically incrustation of live cells. So if I quickly go through to my system here, I have my conditions where they grow with iron, but they're active, they reduce sulfate to sulfide, so they are metabolically active. We also tested incrustation if non-active cells were getting encrusted. So in this case, we take bacteria that are alive, but we remove the lactate from the medium. So there is the possibility of binding iron, but then we do not give them lactate. So basically, they cannot produce sulfide. So in this case, the goal was to add sulfide, but when we left the bacteria for a few days in the incubator, and when we were ready to add sulfide, we saw that the cultures are actually turned black, which was not expected because they didn't have anything to reduce sulfates. And then we actually ended up looking at them before adding sulfide. And so what we saw is that those non-active cells, so them first might have been storing sulfide in their cells and then releasing it in an environment that had no sulfide. And then we can see that the incrustation happens very differently. So it's between the two membranes. So these are bacteria that have two membranes. So basically very different from the other ones. So we have a pattern of incrustation that is different when the cells are non-active. And I will quickly go through those results, but we use scanning transmission X-ray microscopy to look at the distribution of carbon, because we have microbial interaction, we have carbon in the minerals. So this is an image of transmission. This is a mineral aggregate, iron sulfide, sulfur. And then the organic carbon happens as a all over the aggregates, and then we see those hotspots. This is a 2-year-old sample. We still have the cells that are preserved as hotspots, so hotspots of carbon. And then looking at even older samples, so we don't have that kind of resolution for the carbon distribution, but carbon is still there. Carbon, nitrogen, and oxygen. Here we know from spectroscopy that the oxygen is not oxidation of those minerals, it's basically oxygen from functional groups. So we have a preservation of organic carbon as a coating and as hotspots, which has the spectroscopic composition of microbial cells. And we did some quantification. And so in this case, we have experiment with this marine strain and then with a freshwater strain. And then we grew them with iron, with conditions that where they could reduce more and more sulfate. In this case, they produce more and more biomass. So the more biomass is available, the more carbon is stored into the minerals. And so we also looked at, so this is organic carbon in medium and the organic carbon removed from the medium in the minerals. So these are the two bacterial systems. These are abiotic conditions where we added tons of mixtures like yeast extract or a mixture of amino acid. And the bacteria are much better, the minerals like much better the microbial material. And so we can see that the minerals have a higher affinity for microbial organic carbon. So there is a strong affinity between the minerals and the cell material. And so this is for kind of an estimate of the amount of carbon that could be sequestered by those metastable minerals in modern environments. So it is significant. And then I will quickly finish with the final questions, or just a few slides, so we can have time for questions. So we have iron sulfide minerals testing the role of sulfide minerals in the life of bacteria. So this is a representation for those who are not microbiologists. These are the typical life phases of bacteria in a bottle, where bacteria are put in the medium. There is a lag phase. They get used to the medium. They grow, use whatever they can. So they basically produce lots of biomass. Reach the stationary phase. In our conditions, they use all of the supplies available in about 1 to 2 weeks. We can see that incrustation happening, and then we saw that de-incrustation happening. So then it's like death phase. And then there is potentially this long-term preservation of cells at a low cell concentration, that they're very active. They can basically eat their dead friends and survive this way. So my question here was to try to understand, so there's very different aspects that we can look at. What I'm going to show you quickly is I was trying to see if all the organic carbon that is available or stored into those minerals could be available for microbes to eat, basically, when there's no food left available. So basically what we did is that here are the controls. So this is reduced sulfate in different conditions. These are the controls when we give them lactate. So it's their favorite food. They're really happy with it. And then you can look at those experiments from the left to the right. It's just something as a function of time. And the final bar is just the average. So we know that they use lactate in about a week. So that's why we have this variation. It's just based on the way we measure lactate. We measure sulfate. But overall, we can see that they can reduce about 10 millimolar of sulfate in those lactate conditions. And then we did all these different conditions where instead of adding lactate, we added biominerals, so recovered from another culture, and then put into the medium. So they had nothing other than that. We have the biominerals. We sonicated those biominerals. We also use the biomass. So we use like a big pellets of cells that we put in the medium, and then we sonicated it. So we have indication that they can access some of the carbon, but we have to redo experiment here. But it's very interesting that they could potentially be reducing sulfate by accessing the food stored into the minerals to survive. So we are in the process of verifying this, but this would be very interesting. And then, yeah, so to answer the question is that the iron sulfide minerals are likely good for bacteria in this case. And there's other things that we are looking at, but yeah. So to finish my presentation, this is a summary of the potential biosignature that we have. So basically the conclusion is that the biogenic sulfide could be used as a way, basically as biosignature. It's a little more subtle than what I showed you. I didn't have time to present everything, but this is a summary of the biogenic conditions where they're grown with iron, where we basically have a crust forming and then going away. The minerals are larger, they bind to organic carbon, and carbon is very stable. And then we have this greigite formation happening. So what I showed you as well is that the crust could be used as proxy for environmental conditions. So if you have cells, if you find cells encrusted in an environment, it means that they were in active time, maybe. And then we have this metabolic state where they were actively producing sulfide or not actively available to produce sulfide, but releasing whatever they have stored, which I'm still wondering why would they do that. But anyway, when we have the conditions that are where there's not the templating effect, all of this does not happen. So we have smaller size, no incrustation. However, the minerals still store a lot of carbon. So this is very interesting in terms of biosignature still. And then in the what I call the prebiotic conditions, where we precipitate minerals with organic molecules, we have this funny little structure forming mini filaments that are trapping carbon as well. So yes, we might have an issue with the biosignature, but as I said, in those two conditions, we have those hotspots, so cells can actually be preserved like physically for a long period of time. So this is where we have a big difference between the two. And I'm getting late, so I want to thank all the undergraduate students in my group that have produced most of the data, funding, facilities, and I would like to thank you for your attention, and I'm ready for questions. Sorry, I went a little bit over time. Get back to, so I don't know how we do that for questions.
Oh, thank you so much, Dr. Picard.
Yes,
so what I can do is I can read off the questions in the chat, and then if
absolutely.
anyone just wants to unmute their mic, they can do so later. So we have 3 in the chat. So the first one is from Adrian Lopez, and this individual asks, the interaction between some minerals and the enhancement modification of biochemical pathways has been observed in other biogeochemical cycle elements besides sulfur.
Okay, wait a second. Can you repeat the question?
Yes.
Sorry.
So the question is, has the interaction between some minerals and the enhancement, let's see. Sorry, my chat got moved down. Has the interaction between some minerals and the enhancement modification of biogeochemical pathways been observed in other biogeochemical cycle elements besides sulfur?
That's a good question. Okay, so the only one that I know well is the interactions between ion oxidizing bacteria and the formation of oxides. So in this case, other case, as I said, incrustation, when it happens, it is kind of deadly. So cells either avoid being encrusted by changing the cell surface property, and it's very cool. They are type of bacteria that can direct the precipitation of minerals onto extracellular structures, so twisted stalks or sheath. And for the other one, basically incrustation would lead to something that is basically not good. So the cells that are encrusted in oxides, they cannot assimilate carbon or anything that comes from the outside. What is very interesting with the sulfides is that iron sulfide minerals are semiconductors. And so pyrite, for example, is used in solar cells as thin films. So the idea is that microbes, microbial activity, microbial metabolism relies on redox reactions. So everything is very based on being able to access electrons from the inside or from the outside. So this is why I think the iron sulfides are kind of unique. It's because the potentially there would be a way to get those electrons out of the minerals that trap the carbon, or yeah, maybe just enhance it. So not to my knowledge, but there's other system that I'm interested in, like the carbonate system, obviously, where we see different patterns of incrustation and intracellular precipitation. So I think there's still a lot of open question in the field for sure. I hope. Does that answer the question, or did I miss something?
No, thank you.
Okay.
Yeah, I'm sure that individual appreciates the answer. So I just found out that we cannot unmute mics. So if you do have a question, please post it in the chat. So the next question is asking, can the addition of sulfide-bearing amino acids, such as cysteine, substitute for the addition of the inorganic sulfur to the bacteria?
Yeah, that's a good point. So in all of my abiotic system, I tried to, basically the idea now with this spectroscopy, X-ray spectroscopy, is trying to understand if there is potentially covalent bonding between the minerals and the biomass. And so that would have implications about how accessible that biomass is, or those molecules are. I cannot answer now, but I was trying to figure out, so X-ray spectroscopy might potentially tell us about binding between ions. So obviously iron binds to iron binds to sulfur, and that's mostly the binding that we can measure in those minerals. But I was hoping to be able to measure potential binding, or detect potential binding between iron and carbon, or iron and nitrogen, or iron and oxygen in functional groups. Same if we do spectroscopy from the sulfur point of view. So I do have a lot of different controls that have been precipitated with cysteine. It's still in the progress, and I have been time coming up in the summer to look at the minerals from the sulfur point of view. So hopefully this will be answered in the near future.
Awesome, thank you. And then what looks to be our last question is from Mohit Deswani. So they say, thanks for this really interesting talk. Do you think we could use the characteristic particle size of the greigite as a potential biosignature? For example, if a mission to Enceladus were to discover large greigite crystals emanating from the ocean, would you be confident in saying that sulfate reducers were involved in producing the large greigite crystals? Is there a cutoff particle size for biogenic versus abiotic?
Yeah, okay. So would I be confident just by looking at the particle size? No. Obviously, we need to have all these different aspects. So as I said, I'm looking at the crystal size, the carbon content, the composition of the carbon. What I didn't mention in those, because I didn't have time to present all the what I call prebiotic thing, but this is published. So basically, the idea is that when we started with small compounds, so we basically had those abiotic conditions, and then added with iron. So we added those molecules, and then we added sulfide, and then we looked at the carbon using X-ray spectroscopy. So it's not like giving us molecules. The spectra is just an indication of the type of functional group and types of molecule that we have. But when we have those conditions here, basically, we saw a shift in the spectrum, indicating a potential kind of polymerization of those like more simple organic molecule. So it looks like those sulfide, they might be actually really doing this kind of polymerization or helping to link simple molecule to one another. So again, like there is lots of aspects that we have to look at. So this picture, this little diagram is just to show that this is much more subtle. So to finish with the gregae question, gregae can be formed without microbes. It typically involves a little bit of oxygen and specific conditions. There are pH conditions where gregae form very easily, quickly without microbes. So obviously, like any other biosignature, you need a number of parameters that would check the box. Also, I have to add that sulfur reducers change the sulfur isotopic composition of sulfide and can lead to very large variation that are unique to the biological process. However, it really depends on the condition. So I think you really need all of the parameters possible measured before definitely before making a decision about that was formed by life or not. And yeah, don't hesitate to send me an email. You can find my email by just typing my name and UNLV, and we'll get to that if you have additional questions.
Awesome, thank you. And we do have one more question that just
Yes.
popped into the chat.
Yeah, absolutely.
So this question is, do you know if chelated iron is available for oxidation metabolism?
Yes. The thing is that those microbes are not actually oxidizing iron, but yeah, I think for 1 second. In cultures of iron reducers, the iron can be provided as iron to sulfide. It can be, the thing is, it oxidizes really quickly. So it has to be in a kind of stable form. So some iron oxidizers can get electrons from iron too, but they also need like low concentration of oxygen. So they kind of compete with the chemical oxidation. So typically, yeah, it would be stabilized as iron 2 chelated by organic ligand, for example. And I don't think it's an issue for microbes. It's actually probably even better because it's already just like they just have to grab it from the ligand versus from a mineral. So it is possible for iron oxidizers. Yeah.
Awesome, thank you so much.
Thank you to everyone
And
here.
with that, we'll conclude today's April session for the Life RCN seminar series. Thanks everyone for joining us. And thank you, Dr. Picard.
Thank you so much for everyone coming, and thank you again for the invitation. It was a pleasure. Bye, everyone.